Hello, my name is Lars Westblade, and I'm from Weill Cornell Medicine. In this video, I want to tell you about vancomycin-variable enterococci. In order to do that, I need to tell you a little bit about the target of vancomycin, and that is the bacterial cell wall, also known as peptidoglycan. Bacterial cell wall forms this mesh-like structure that provides structural integrity to the bacterium. If you inhibit the cell wall synthesis, cells will lyse because they have lost that structural integrity. Vancomycin is a glycopeptide because it's composed of carbohydrates and a peptide. It is only active against gram-positive bacteria, and that's because it cannot cross the outer membrane of gram-negative bacteria. Vancomycin inhibits cell wall synthesis by sterically excluding enzymes, specifically transglycosylase and transpeptidase enzymes, that are important in making that mesh-like peptidoglycan cell wall. An important mechanism of vancomycin resistance is that mediated by the vanA, vanB genes. If we consider a vancomycin susceptible Enterococcus faecium, the cell wall looks like this in the cartoon, where we have dangling down two dealanine residues. Vancomycin can bind to these dealanine residues and therefore sterically occlude the binding of transpeptidase enzymes that are involved in cross-linking the cell wall. As I said, if the cell wall synthesis is inhibited, you get cell lysis. If we have a vancomycin-resistant Enterococcus faecium, and the resistance is mediated by vanA or vanB, that last dealanine residue is substituted with a D-lactate residue. That simple substitution prevents the binding of vancomycin to the cell wall, and so therefore it cannot sterically occlude the activity of the transpeptidase and transglycosylase enzymes. So cell wall biosynthesis is not inhibited, and there is no cell lysis. The vanA vanB mediated vancomycin resistance mechanism is inducible. So what does that mean? Ultimately, what it means is that vancomycin can turn on the genes that encode the expression of enzymes that confer resistance to vancomycin. So vancomycin is sensed by a protein called vanS. vanS phosphorylates itself. It can then transfer that phosphate group to a protein called vanA, which is a transcriptional regulator. The phosphorylated vanA protein is then activated and combined to the DNA upstream of the vanHAX operon and turn on the genes that will confer resistance to vancomycin. Those genes, or the products of those genes, will convert the formation of diala that's associated with the cell wall to diala D-lactate. So there's this target modification, and this modification reduces the affinity of vancomycin for the cell wall 1,000-fold. There's a very interesting phenomenon known as vancomycin-variable enterococci. These are isolates of Enterococcus, typically Enterococcus faecium, that test vancomycin susceptible, but they encode the vanA gene. And they've been described in both North America and Europe. And in fact, in one Canadian study, it was shown that vancomycin-variable enterococci comprised almost 50% of vanA-positive Enterococcus faecium isolates that were collected from sterile sites. In these vancomycin-variable enterococci isolates, vancomycin does not induce vanA expression. And that's because there's been this decoupling of vancomycin sensing mechanism to the expression of the vanHAX operon. But interestingly, and rather concerningly, through vancomycin exposure, vanA in these vancomycin-variable enterococci can be turned on and become vancomycin-resistant enterococci. And there are various mechanisms that lead to this. It's a concern that vancomycin-variable enterococci can readily disseminate undetected, as most laboratories do not test enterococci using vanA vanB molecular methods, with the notable exception of positive blood cultures. So we really don't know how big the issue of vancomycin-variable enterococci is. However, if you are to detect one in your laboratory, you should report them as vancomycin-resistant enterococci. And this gives me an opportunity to encourage you all to match up phenotypic and genotypic results. So that is, if the phenotypic result does not match up with a molecular result, you should always try and resolve it and troubleshoot it. And then, fortunately, we have guidance that's provided in the form of the CLSI M100 document, notably the Appendix H, using molecular assays for resistance detection. So when we troubleshoot, we should always ensure that we're working with a pure isolate. So look at the purity plate. We should consider confirming the identification, confirm the initial testing results, and ultimately make sure that there were no clerical issues. Finally, if resolution is not possible, it is advisable to report the more resistant result. Thank you so much.